Hello, ladies and gentlemen. Welcome to the sixth session of How I Treat Autoinflammatory Disorders course. I am Dr. Kosar Asnashari, Assistant Professor of Pediatrics at Children's Medical Center, Tehran University of Medical Sciences, Iran, and I'm the head of the executive team for the course. This course is programmed as a progression to How I Treat series. How I Treat series are educational programs planned by the leading house for Iran North America academic partnerships. Each of these programs try to provide information on basic knowledge and clinical experience related to treatment of a disease or an aspect of a disorder. Our course, How I Treat Autoinflammatory Disorders, includes 9 sessions in which we discuss different aspects of autoinflammation, including pathogenesis, diagnosis, treatment, patient education, and network development. Our goal is to bring physicians, pediatricians, immunologists, and rheumatologists with the latest updates on autoinflammatory disorders by the help of novel experts from different nations, USA, Germany, Turkey, Italy, and Iran, as our speakers. In the light of these efforts, we hope a better understanding of the mysterious autoinflammatory disorders can be reached, leading to the rescue of the affected patients. I want to take a moment here and thank Dr. Wahid Ziai, head of our scientific committee, professor of pediatric rheumatology at Children's Medical Center, Tehran University of Medical Sciences, and the president of Pediatric Rheumatology Society of Iran for his full support of the course. We are also very grateful to Dr. Marjan Yaghmai, head of leading house for Iran North America academic partnerships, for her kind support. I would also like to express my gratitude towards Professor Someira Farman Raja, Professor of Rheumatology and convener of APLAR Pediatric Rheumatology Special Interest Group. We are grateful for her kind support, which made it possible for our course to be endorsed by APLAR. Asia Pacific League of Associations for Rheumatology, or APLAR, was established in 1963 in Sydney. The well-known association of APLAR aims to spread and strengthen rheumatology endeavors in the Asia-Pacific region. APLAR's mission and goals focus on providing advanced care to patients with arthritis and other musculoskeletal diseases through the continuing professional development of members, increasing the awareness and understanding about rheumatic diseases, patient advocacy and empowerment, and encouraging research in the field of rheumatic diseases. If you wish to receive a certificate for each session of our course, you need to register via Appraise to Raise website, and the link can be found here in my slides. You can also register for the whole course and receive a certificate of attendance in an international autoinflammatory course approved and validated by APLAR, Pediatric Rheumatology Society of Iran, Iranian Rheumatology Association, and Tehran University of Medical Sciences. For those who were not able to attend our first sessions, there is a possibility to view the video of the session on the website, answer to the post-test, and receive the certificate of the session. If they attend the rest of the sessions and pass the post-test, they're eligible to receive the course certificate. Today's session is entitled Interferonopathies with a special aspect on Rare Proteasomopathies. Our elite speaker of today's session is Dr. Elke Krüger. Dr. Krüger is a full professor of medical biochemistry and molecular biology. She's the director of the Institute of Medical Biochemistry and Molecular Biology, University of Medicine Greifswald, Germany. She has a PhD in microbiology from Department of Microbiology, University of Greifswald, Germany. She is the chair of the faculty MD, PhD, and PhD program and elected member of the faculty board, University of Medicine Greifswald, Germany. Her main research interests are proteostasis controlled by the Ubiquitin-Proteasome-System and proteasomopathies. The moderator of today's session is Dr. Maryam Nourizadeh from Iran. Dr. Maryam Nourizadeh is an immunologist and an assistant professor in the Immunology, Asthma and Allergy Research Institute, or IAARI, Tehran University of Medical Sciences, Tehran, Iran. Dr. Nourizadeh is the supervisor of HLA typing lab and the director of unrelated hematopoietic stem cell transplantation registry of IAARI, located in Children's Medical Center, Tehran, Iran. She is the managing editor of Iranian Journal of Allergy, Asthma and Immunology, and Iranian ISI PubMed journal in the field of allergy and immunology. Dr. Nourizade's research interests include newborn screening of primary immunodeficiency diseases and using TREC, KRAC assay for early diagnosis of T and B cell effects, especially SCID, HLA typing, and tumor immunology. We will now begin this session. I want to ask you, our dear participants, to share your ideas and ask your questions in the chat forum whenever you want. Dr. Nourizadeh, we are all ears.
Okay, thank you very much. Thank you for inviting me as the moderator of this session. And thank you very much from the International Affairs of Tehran University of Medical Sciences, especially Professor Ziai, for preparing these kind of courses, very special courses. Thank you very much for thank you very much from Dr. Krüger for accepting our invitation for this special talk. And I think this talk would be very special because of the topics that has been selected. Interferonopathies is a very new brand term in scientific literatures, as you know, from the early 2000 years, and also proteasomopathies is so interesting topic as it is a kind of monogenic autoinflammatory diseases. I think it's so similar to the primary deficiency disorders, and have lots of questions about that. Thank you very much, Professor Krüger, and we are eagerly waiting for listening to the talk. Please start. Thank you very much.
Yeah, thank you again for inviting me to this interesting session. I will start with screen sharing now. Second. Here we go. And first of all, I would not only thank the organizers for inviting me, I would also like to introduce where we are located. I had a really long time at the Charité in Berlin. But 5 years I moved back to my study location, which is here at the Baltic Sea. I will go for laser pointer, sorry. And we are of course close to Scandinavia, to Denmark, and also to Sweden, which is up here. I just want to show you this location because we are not that famous, such as Harvard or whatever, but we have a very old university which was founded in medieval times. We have a medieval downtown, a very nice university building and a town hall, and we have beautiful beaches, which is worth to live here. After this little lesson to German geography, I would come to my lesson, and I will start with an introduction about interferons and type 1 interferon regulation. Then I will introduce to you the Ubiquitin-Proteasome-System, a special aspect on proteasome regulation and proteasome dysfunction. So as you know, I'm not a clinician, I'm a basic scientist, and we are pretty much interested in this part here, and stumbled then about proteasome dysfunction, about this recently discovered diseases which are called type 1 interferonopathies. And here I will talk about a little bit diagnostics and a few words to treatment. And then I will go to very recent data on rare proteasomopathies. And I have to make you aware that we have 2 clinical entities. We have this PRAAS phenotype, which is a proteasome associated autoinflammatory syndrome. And we have another syndrome which causes neurodevelopmental delay. And at the moment, we have no idea why. You all know that interferons are cytokines of the innate immune system, and these cytokines are normally there to fight against viruses. So our current pandemic, of course, make us aware of that, and a couple of pattern recognition receptors detect intracellular pathogens via pathogen-associated molecular patterns with respect to viruses. This is mainly nucleic acids. And then they start to produce chemokines and cytokines. So interferons are cytokines, of course, that have a pro-inflammatory downstream mechanism to recruit, to activate other immune cells, to induce adaptive immunity, and to recruit other immune cells. But you should be also aware that damage-associated patterns are also recognized by the innate immune system via pattern recognition receptors. And this will be important in the next slides. We have mainly three types of interferons, type 1, 2, and 3. Type 2 interferons are classical interferons made by T cells when they are activated, but also by NK cells. This is mainly known. The type 3 interferons is mainly interferon lambda, which is also made by many cells, but is not investigated. In this detail, we have here and today a lesson about type 1 interferons, which mainly include interferon alpha and interferon beta. And these interferons are produced by many cell types, also non-immunological cell types like fibroblasts or endothelial cells. As shown or depicted here in the scheme, a virus-infected cell produces interferons, and this interferon signaling, which is sensed by type 1 interferon receptors, then causes signals to neighboring cells to put up barriers. They can cause cell death, but they can also recruit other immune cells and finally start to induce adaptive immunity. Intracellularly, we have the detection of mainly nucleic acids, and in the case of type 1 interferons, this is mainly pathogen-derived nucleic acid, like DNA or RNA, with a myriad of pattern recognition receptors. The endogenous toll-like receptors are sensors for nucleic acids, but also these kinds of RIG-like receptors, or these SIGAM STING pathway, which senses DNA. And there are a couple of other DNA sensors, which then activate the downstream pathways, mainly kinases like TBK1 or IKK, or these MIDI88 pathway, which is engaged by the toll-like receptor signaling. And these kinases then phosphorylate transcription factors, mainly IRF3 and IRF7 in the type 1 interferon signaling pathways, but also NF-kappa B can be activated by phosphorylation of I-kappa B, which then releases the NF-kappa B into the nucleus. This leads to transcriptional activation of type 1 interferon genes, interferon alpha and interferon beta genes, which then produce these cytokines, which are secreted and can via autocrine or paracrine loops induce the type 1 interferon IFN receptors, which then activate JAK-STAT signaling. So a Janus kinase activates a STAT transcription factor, which then goes into the nucleus and amplifies this loop by induction of interferons and interferon-stimulated genes. Please keep in mind interferon-stimulated genes, because this is a mechanism how we detect interferon signaling in a patient. Of course, interferons are pro-inflammatory cytokines, and the pathways have to be tightly regulated. This is again summarized here. We have these myriads of nucleic acid receptors, and the pattern recognition results in signaling, and the signaling is then amplified by positive regulators or restricted by negative regulators. These signaling kinases and transcription factors are, of course, positive regulators, as well as the IFN receptor or the Janus kinases or the STAT transcription factor, which comes from this side here. And we have a couple of negative regulators which restrict the system. And among these negative regulators, there are many DNA and RNA processing factors, phosphatases, a couple of ubiquitin factors, and among these are ubiquitin ligases, the proteasome itself. And this is important to know why the proteasome is involved in this pathway, because the ubiquitin-dependent degradation of such positive regulators here restricts finally interferon signaling. Too much or too less interferon is bad and is normally causes disease. And as we know from the pandemic now, we have a couple of virus-induced cytokine storms, not only by SARS-CoV-2, but also by, it's known by influenza and other viruses. There are a couple of treatments where they use interferons, for instance, in multiple sclerosis, which can cause interferonopathy-like symptoms. We have the type 1 interferonopathies, but also cancer is coupled to dysregulation of this pathway, where mainly Janus kinases are activated in cancer cells to promote survival of the cell. And of course, too less is bad, because then we have immunodeficiency. Now I would like to introduce to you the Ubiquitin-Proteasome-System. And of course, you know that ubiquitin is coupled to protein substrates via a 3-enzyme thioester cascade. So biochemically, the E1 enzyme, which is the ubiquitin activating enzyme, takes ubiquitin as a small protein. And in energy-dependent manner, this ubiquitin is activated, forming a thioester bond, and transferred to E2, which is a ubiquitin conjugating enzyme. And this E2 enzyme, together with a substrate-specific E3 ubiquitin ligase, then conjugates ubiquitin to protein substrates, which are then, after a couple of rounds of ubiquitin modification, making these ubiquitin chains, target these proteins for degradation by the proteasome. This process is important for cellular protein homeostasis, or shortly proteostasis. The ubiquitin-proteasome system is responsible for protein quality control and homeostasis of almost all types of proteins in cells. So they are cytosolic, nuclear proteins, ER or membrane proteins, which are degraded by the ER-associated degradation system. This degradation system controls the availability of regulatory proteins such as transcription factors, but also cyclins. So if the proteasome does not work, the cell cycle progression is inhibited. And therefore, the system is directly involved in signaling and regulation of transcription. And the ubiquitin-proteasome system generates ligands for the MHC class I antigen presentation and is therefore directly involved in adaptive immunity. So my group is interested in the regulation of the system, and we are interested how different proteolytic requirements can be met by adaptation of cellular programs, and the consequences of such adaptation processes in health and disease. And of course, if the proteasome does not work, this is known for years that this is involved in neurodegeneration and aging. However, the involvement in the innate immunity is less defined, and therefore we are working in this topic. We started years ago with the regulation of the proteasome as a enzyme complex. You can see that we have sophisticated assembly processes to build up these 26S proteasome complexes, which are mainly composed of the 20S proteasome core complex containing the active sites, which are here in these inner beta rings. During the assembly process, these are coupled to 19S regulators, which are important for recognition of ubiquitin substrates, for unfolding of these ubiquitin substrates to feed in the substrates into the proteolytic chamber here. And we know that this process is regulated by gene expression. So the subunits undergo gene expression regulation. We have epigenetic mechanisms. The assembly process is assisted by helper proteins. We have discovered this guy here, which is called proteasome maturation protein, or shortly POMP, which helps to integrate the subunits into these regular ring structures. The assembly process may incorporate alternative subunits to form alternative complex structures, and the association of regulator complexes is also important. And in the course of interferon signaling, we have a faster complex assembly. So the cells adapt by a quick assembly of alternative isoforms, which are called immunoproteasomes. They have alternative active sites. They also have alternative regulators, and so-called immunoproteasomes or hybrid proteasomes confer higher proteolytic activity to the cell, and also altered proteolytic activity to better meet the demand of antigenic peptide generation. As I said, protein homeostasis or proteostasis is very important for cells, and you see that this homeostasis is a sophisticated balance of protein synthesis, protein quality control, and protein degradation. For protein degradation, I already introduced to you the major non-lysosomal degradation system, which is the Ubiquitin-Proteasome system. And we have, of course, the lysosomal degradation. And here, the intracellular degradation system is autophagy. And these systems, of course, crosstalk and cooperate with each other. I will show you also an example of that. Imbalances cause diseases. However, cells can counteract these imbalances by upregulation of protein quality control and degradation systems, or by shutdown of translation. And major cellular responses are the unfolded protein response or the integrated stress response. I will come to that later because this is one of the mechanisms how we think interferon production occurs in response to proteasome dysfunction. We know that inflammatory conditions, interferon signaling, also toll-like receptor signaling or interleukin cytokine signaling, but also neurodegenerations or pathogens damage proteins already at a level of translation, which is shown here. We detect higher radical production or ROS production in cells, and this can also occur in the course of proteasome impairment. When you inhibit the proteasome, then the translation process is challenged by ROS production, and the cells have to react somehow. And we have shown this by different means. In the past, we have used animal models where immunoproteasome activity is deficient. And in these mouse models, we performed a multiple sclerosis model. And you can see here, these green dots is ubiquitin staining. The neurons accumulate such ubiquitin aggregates, and also the disease scores are higher in immunoproteasome deficient mice. And we could show a similar pattern infecting mice in a Coxsackie virus B3 myocarditis model. These are heart sections here. This is a brain section. And you can see here also with these green dots that we observe ubiquitin accumulation and a delayed resolvement of disease and higher disease progression in immunoproteasome deficient mouse models. Similar pattern are observed in such proteasome-associated autoinflammatory syndromes. This is a skin section where we, together with the group of Rafaela Goldbach-Mansky, stained ubiquitin. And of course, when the proteasome does not work, one would expect the accumulation of ubiquitin aggregates. And this is a kinetic thing. Of course, if there is still proteolytic activity, these aggregates can be removed. The same things you observe in neurodegeneration. We also have neurodegeneration models used, and I will show you an example a little bit later. We observe interferon-induced cell death in these mice. which are deficient for immunoproteasome function. And in patients, we observe interferon production. We also observe this in mouse models, and we do see altered gene regulation. Here is the activation of NF-kappa B, which is impaired in immunoproteasome deficient settings. And this is a quite complex slide, but I will try to guide you through. So we start with an impaired proteasome activity. And I have shown you that then, as a response, cells accumulate ubiquitin aggregates in the cytosol. However, the proteasome is also responsible for degradation of proteins of the secretory pathway, membrane proteins, ER proteins, proteins of the Golgi, or lysosomal proteins, which are quality controlled here in the ER, and proteins which do not meet protein quality control are retranslocated out of the ER and degraded by the ER-associated degradation system. So they are mainly ubiquitin ligases located here in the ER membrane, which ubiquitinate these proteins and feed them into degradation by the proteasome. When the proteasome does not work, ubiquitin-dependent protein defective proteins will also accumulate in the ER and alarm these unfolded protein response mechanisms. And the transcription factor, which is directly coupled to the ARID system, which is called NF1 or TCF11. We have discovered also years ago that this transcription factor is activated in response to proteasome impairment. The activation occurred via a protease, which is called DDI2. And the cleaved transcription factor translocates into the nucleus to activate proteasome gene expression. So this mechanism is a rescue mechanism in response to proteasome impairment. The unfolded protein response, as well as the integrated stress response, here abbreviated with ISR, is a mechanism that stops or arrests translation or protein synthesis by phosphorylation of this translation initiation factor here, EEF2 alpha, and a couple of EEF2 alpha kinases is responsible for that. This is the protein kinase PERK, which is located in the ER membrane. Protein kinase R, which is a cytosolic protein, and a GCN2, which is kinase activated by viral infection, by interferons, but also by double-stranded RNA. And this kinase is activated by amino acid deprivation. Downstream of this translational arrest, there is the activation of a transcription factor called ATF4. And ATF4 in turn activates genes for autophagy, for apoptosis, proteins involved in lipid or amino acid metabolism, protein quality control system, ARID system, and so on. There are two other arms of the so-called unfolded protein response, which senses protein misfolding in the ER, which are ATF6, which is a transcription factor on its own. This transcription factor is also activated by proteolytic processing in the Golgi apparatus, which then releases the transcription factor into the nucleus. But we also have this IRE1 alpha system, which is a kinase and nuclease system that can degrade mRNA, which accumulates in response to this translational arrest. And it can activate this transcription factor here, XBP1, by a certain kind of splicing, which then releases spliced and activated XBP1 into the nucleus. So all these stress response systems integrate these proteostatic imbalances in cells and try to rescue this by activation of rescue factors such as protein quality control and degradation factors, but also metabolic factors. The final or ultimate response is, of course, cell death. And all these responses are somehow activated in response to proteasome dysfunction. Now I will come to the point of interferonopathies and diagnostics and treatment. And we have major clinical entities which are comprised by the so-called type 1 interferonopathies. This is the Aicardi-Gouttier syndrome with a major neurological manifestation. This is a syndrome which is called Kendall. The more recent description is in this abbreviation PRAAS, proteasome-associated autoinflammatory syndromes. Kendall describes a little bit the symptoms of this disease with chronic atypical neutrophilic dermatosis with lipodystrophy and elevated temperature. We have a broad spectrum of symptoms, including neuroinflammation. And another clinical entity is SAVI, the sting-associated vasculopathy, with onset in infancy with a peripheral manifestation. And all these syndromes have in common that these patients suffer from dysregulation of type 1 interferon signaling. So the strict regulation of type 1 interferon signaling pathways is somehow dysregulated, and therefore these patients have autoinflammatory syndromes. Again, I will come to this slide because this explains how the genetic courses of these diseases can explain the clinical phenotype. So on one hand, we have gain of function mutations in positive regulators of interferon signaling, and we have loss of function mutations in negative regulators of interferon signaling. And I will go into detail here now by showing the current major mutations which are known. This is a this figure was taken from a review by Yannick Rowe and Daniel Stetson, recently published in Nature Reviews Immunology. And I think this picture here summarizes the major genetic deficiencies here. We have a couple of gain of function mutations in STAT1 and JAK1, which are positive regulators. These are post-interferon receptor signaling factors. We have loss of function in negative regulators, such as ISG15, USP18, or STAT2. We have loss of functions in a couple of proteasome genes, which are negative regulators in interferon signaling. without knowing a detailed disease signaling failure here, but we have an idea now. We have a couple of unclear genes, including these complement factors here, and this lysosomal phosphatase. Genes which are involved in mitochondrial integrity are important, such as NGLY1 or ETA-D3A. We have a couple of factors which are involved in nucleic acid metabolism and nucleic acid sensing or nucleic acid signaling. Many of them are known disease-causing genes in Aicardi-Gouttier syndrome. We have STING as a major gain of function mutation in SAVI, or the COPA syndrome, which is a factor involved in STING intracellular trafficking. So this is a factor which is involved in cellular trafficking. And we have these nucleic acid sensing factors, which are also involved in type 1 interferonopathy. Clinical entities. Here, this is broken down a little bit in more detail into cellular locations. We have these nucleic acid processing factors, which inhibit the production of these RNA-DNA hybrids, which are sensed by these nucleic acid receptors. The most important message here is that also self-DNA can be sensed by these factors here, including RNA sensing by RIG-like receptors. This is mainly mitochondrial DNA, which can be released by mitochondria when mitochondrial integrity is somehow affected. This is clear when these genes NGLY1 or PNPT1, PNTP1 is mutated, then the mitochondrial integrity is impaired, and mitochondria release mitochondrial RNA and DNA. And this can be sensed by the RNA and DNA sensors. So we have DNA processing factors which are involved in these diseases. We have RNA processing factors which are involved in these diseases. We have signaling factors such as USP18, which is a negative regulator of JAK-STAT signaling, and when mutated, it causes a pseudo-torch syndrome. We have the proteasome, which somehow is a negative regulator of ISG production, probably by inhibiting these signaling pathways. IRF3 is a known substrate of the proteasome. The same is true for MAFs and also other factors, the STAT factors, of course. We have these Artemis and ATM systems, which can cause DNA damage. And all these loss of function or gain of function mutations are known causes of type 1 interferonopathies. So as soon as you have a new mutation and you look in a factor which is involved in these signaling pathways, it's clear that you can have a look into type 1 interferon production in the patient when there is a clear sign of autoinflammation. This slide somehow depicts also other diseases which fall into this category. We have this monogenic systemic lupus patients, other factors which can be caused by, or other clinical entities which can be caused by nucleic acid processing factors. Here again, Aicardi-Goutier monogenic SLE, or the so-called Singleton-Merten syndrome, can be caused by mutations in these sensing factors here. We have positive interferon signaling regulators such as STING. We have the proteasome genes. We have these complement factors, which are known defects in monogenic SLE. We have this kind of lysosomal phosphatase factor, which is also involved in bone cell regulation and calcification. And we have these interferon signaling factors, which are known in other clinical entities, such as the pseudotorch syndrome. When we go into the proteasome system, it's important to know, and we have published this in a review, and we are trying to repeat these in several other reviews as well, that we have roughly 50 genes which encode components that contribute to proteasome to proteasome complexes or encode proteasome assembly helper proteins, such as these PEG factors, which are encoded by genes called PSMG1 and 2 and 3 and 4. We have a POMP, which is also a known mutation in the known cause of PRAAS. PSMG2 is also a known cause of PRAAS. And you can see that we have a variety of complexes which can be built, and theoretically, mutations in all of these 50 genes here can lead to so-called proteasomopathies. And therefore, not only the major founder mutations, which are mainly located in the immunoproteasome, which is depicted here in the subunit PSMB8, are important. Please be aware, this is not an immunoproteasome disease, this is a proteasome disease. And potentially 50 genes can contribute to this degradation complex in different isoforms. And at the moment, we have we have 10 mutations which are published, or 10 subunits with published mutations. We know another 10 from databases and our collaboration partners. And as I said, potentially 50 genes can be involved. And this is an entire gene panel which has to be taken into account here. This is important for diagnostics, and therefore I'm saying this. Of course, we have clinical diagnostics. This is a picture of a classical Kendall patient or PRAAS patient with this skin lesions here. Histologically, you see this infiltration of neutrophils into these skin lesions. We have chilblain lesions and necroticism and vasculitis as a clear sign of autoinflammation, interstitial lung disease, paniculitis, or lipodystrophy. At the moment, the course of lipodystrophy is a bit unclear from the molecular part. We have a disease in the joints, of course, polyarthritis, early thrombotic events. We have kind of cytopenia in many cases. We observe autoantibodies, such as the typical SLE-like autoantibodies and other inflammatory signs. This can be coupled with classical CRP or interleukin induction. For the clinical entities with a brain manifestation, you can see such basal ganglia calcifications here. So it's also important to look into brain MRIs or CTs to find these brain abnormalities. And along with the clinical diagnosis, there is a special need for the genetic diagnosis. And in terms of the proteasome, we do not only have monogenic diseases. As I said, we have 50 genes which can contribute, and we observed a couple of digenic inheritance. I depicted 2 examples here, where we have compound heterozygous mutation in one family and a digenic mutation in another family, where we have a mutation in one subunit encoding an alpha subunit here, and another subunit which is the classical Kendall founder mutation, PSMB8. We have a couple of de novo mutations which are meanwhile known. We have heterozygous and homozygous mutations. And therefore, I'm saying it's not only monogenic. This is important. All these diseases have a common molecular phenotype, and this is the increased type 1 interferon signature. As an example, I used this from one of our publications together with Rafaela Gold-Bachmansky. This is a classical RNA sequencing experiment where you more or less sequence the entire transcriptome, and then you see the differences between the patients and the parents and the healthy controls. And red means upregulation, blue means downregulation, and you can clearly see a distinct pattern. And all these genes here are classical interferon stimulated genes which are upregulated in the patients, but not in the controls, and also not in the often heterozygous carriers or among the parents of or relatives of the patients. In terms of proteasomopathies, it's necessary to run also functional assays for the proteasome to define that the mutation is pathogenic. And I will come up with a couple of examples how to diagnose this, how to measure an interferon signature. So here we measure interferon stimulated genes. To measure directly the cytokine is difficult. It works a little bit, but it's difficult to perform in the clinical chemistry because these interferons are very short-lived. However, the transcriptional patterns which we observe here, these interferon scores, these are a little bit clear, more clear than the cytokine pattern. So we indirectly measure interferon upregulation. Normally, blood samples, PAX gene blood samples are used for that to isolate RNA. This RNA is then used for cDNA synthesis. with a classical qPCR analysis using 6 gene panel, or you use the nanostring chemistry, which is shown here. You have a kind of hybridization method where you have these barcode probes which hybridize to your target sequence, and then you can see, can you read out these barcodes, giving special patterns. I depicted 2 of such examples here. Here you can see a work or an example using Aicardi-Gouttier syndrome patients, which we investigated together with Thilo Kalinich from the Charité in Berlin. They have this clear interferon signature using the median of these ISGs here, IFI44L, IFI27, ISG15, RS82, cyclic 1. And this ISG pattern should be calculated against a couple of healthy controls. The more, the better. And then you can calculate these scores. Scores above 2.54 are designated as significant. And with this group, they also developed a flow cytometry method where they stain cyclic 1, which is a surface marker of monocytes. And this is also a clear sign when you investigate patients' blood and stain cyclic 1 or CD169 at the surface of monocytes, then you have a quite reliable, quick method to see whether you have an interferon signature or not. This is an example of nanostring here, where you can use customized panels, but you can also buy commercial panels by this company. We use the commercial panel here, which is the autoimmune profile panel. And you can clearly see red means upregulation, green means downregulation or normal. A clear interferon signature, all these genes here are upregulated in two candle patients. This must be candle 1 and candle 2. I'm sorry for this mistake. Just to give you an idea how this looks like and how this pattern works here. Here I have depicted how we do this. We collect whole blood samples, and it's important that you need PAX gene blood for transcriptomics. When you send blood to big centers, it's also nice to have 1 vial of EDTA blood, because here you can produce viably frozen peripheral blood monocytes from that. The plasma can be retained for further investigation of the plasma. From the PBMCs, you can produce stem cells, induce pluripotent stem cells. You can make cryostocks of them. We produce T cells from them, because then we have a lot of cell material to do a downstream analysis. This is important for doing biochemistry. When we do biochemistry with that, we need more cells than 1 blood vial. And to measure the activity of proteasomes, we have different methods, but a clear clinical sign of proteasomopathy is the accumulation of ubiquitin and the impairment of proteasome activity. For proteasome activity, there are mainly 2 basic methods. One method measures peptide hydrolysis. Here we use fluorogenic peptides. and feed them into the proteolytic complexes. You can read out this via plate reader or via a native page analysis. Here you have different cells. So this is a control, this is a candle patient. And you see that the proteolytic activity and the peptide hydrolysis activity of the 20th proteasome or the 26th proteasome here are impaired. And there is another method where we use so-called activity-based probes. This is a peptide which fits into the active site. So the protease reactive element, this is the warhead here, and we have a retrieval element. In this case here, we use a fluorophore as a tag. And these activity-based probes are mainly peptide inhibitors, which are coupled to a fluorophore. And here we can measure the accessibility of active sites. And this can be monitored by different readouts. You can see this in microscopy, in a plate reader assay. You can do gel-based assays, but you can also do FACS flow. And this is easy to measure, and we will develop a proteasome assay based on this activity-based probes, which will be made available to all of you when we are ready with our investigations and testing of these activity-based probes. To have a little excursus into a treatment. At the moment, we are talking about the advent of anti-interferon therapies. And you all may have read the papers which apply JAK inhibitors for the treatment of autoinflammatory diseases. There are a couple of them available, and they work quite well in case of proteasome-associated autoinflammatory diseases. Patients with SAVI also respond to JAK inhibitors. Aicardi-Goutier patients do not respond very well because of the pharmacokinetics and dynamics of these inhibitors. They do not cross the blood-brain barrier, so it's a little bit harder to treat these patients. But this is not my expertise, so here I refer to Yannick Crow, who is an expert in that. And recently there was this IFNAR antibody released, anifrolumab, for rheumatologic diseases. This antibody blocks the IFNAR receptor, and thereby the made by the antibody, by the interferons. Together with Cecilia Poli and Taku Kuipers, we also investigated patients which have received a hematopoietic stem cell therapy. Here, it's important that this is an curative treatment possibility. So these patients are resolved now from infection and autoinflammation. But we also use this to establish that viably frozen PBMCs allow retrospective analysis of patients. And T cells, which are expanded from these PBMCs, allow to recapitulate the molecular phenotype. We do see interferon scores before HCSC treatment, and this is gone post treatment, and the same is true for the activity of the proteasome. So this is a PSMB4 patient, but we also investigated 2 patients with mutations in POMP with Cecilia. And here you can see before treatment, these T cells have much less proteolytic activity compared to healthy controls than the patient. The patient has less activity, and after treatment, also the proteolytic activity is recovered. And finally, I would like to introduce you my co-worker and postdoc, senior postdoc group leader, Frederick Epstein, just to remind you that proteasomes are involved in signaling, and that inhibiting the proteasomes by proteasome inhibitors, which are used in cancer, but also with knockout mice and in these rare proteasomopathies, kill all these signaling pathways, and this can be observed in patients that they are altered here. Again, we have two clinical entities, and I try to hurry up a little bit. I see that time is running. So we have this neurodevelopmental delay phenotype. We have clear effects in developmental pathways. You can see here that there is developmental failure in the development of fingers. They also come up with these chilblain lesions, but in the systemic inflammation, you see the inflammation on just looking at the patient, but the molecular phenotype is the same. Both clinical entities accumulate ubiquitin and show impairment of proteasome activity. So this has been done with this pan-reactive activity-based probes, and we hope to come up with this method soon. And both types of patients show up with higher interferon scores compared to controls, and this can be recapitulated in T cells. But using nanostring analysis, we observe different molecular patterns in the transcriptome. We have specific patterns which are specific for this NDD phenotype in comparison to the PRAAS phenotype, and we are currently investigating what is going on here. But the red block here, which is common to both, is the type 1 interferon signature. And again, although these patients suffering from neurodevelopmental delay do not have inflammatory signs at the first glance. So skin lesions and other inflammatory signs are rare here. So they have clear brain manifestation and developmental manifestation. Just to remind you that these pathways play a role, we have investigated these pathways in the patient settings, and this is exemplified here by a patient with PSMD12, which we have published recently. You can see the activation of this transcription factor, which activates proteasomes, so the cells try to compensate for proteasome impairment. We do see accumulation of ubiquitin, which is not shown here. We do see the upregulation of the integrated and unfolded protein, integrated stress response, unfolded protein response here. We do see the impairment of mTOR and the activation of mitophagy, which is shown here. Sorry, in this slide, in the scheme. So the mTOR downstream pathways are impaired, and this can be also seen in neurodegeneration models, which do show upregulation of type 1 interferons. This is interferon beta here in Alzheimer's disease setting. also in the course of aging. And we observed this when we use Velcade as or Proteasomab, which is the name of the substrate, which is used for treatment of multiple myeloma. And we can induce interferon signatures in microglia. And this can be blocked by inhibitors of the IRE1 alpha subunit in the IRE1 arm of the unfolded protein response. So this is clearly involved in this signaling cascade here. And I have already mentioned that we do see the induction of autophagy, which can be seen here. We have much more mitochondrial staining in the lysosomes. And this is a sign of impaired mitochondrial activity and integrity. We are deeper investigating this at the moment. And we do observe the involvement of the integrated stress response. And this, these are my last slides. We have done this work together with the group of Seth Masters in Australia. We made them aware that PKR is one of the kinases responsible for phosphorylation of EF to alpha and translational shutdown. And they investigated the interactors of PKR and found that IL-24 is a major interactor in the course of proteasome impairment. So PKR can be inhibited by this inhibitor, and this is this publication appeared recently, and Sophia Davidson is the first author of this manuscript. They use different patient samples to inhibit PKR activity, and you can see here that interferon beta levels are downregulated transcriptionally. This is not true for healthy controls or for SAVI patients, and the same is true for the interferon score, which is downregulated in the patients, but not in healthy controls and SAVI. And this can be recapitulated in THP1 cells. This is a monocytic cell line, which are genetically engineered for PKR knockout. So PKR knockout or treatment with, so this is a proteasome inhibitors, and treatment in knockout of PKR does not show the upregulation of interferon signature. And the same is true for IL-22 knockouts. And therefore, we came up with this scheme. So IL-24, which is a cytokine which goes the secretory pathway, accumulates when the proteasome function is not is disturbed or perturbed. Then this accumulated and aggregated interleukin-24 is sensed by PKR, which can induce IRF3 and a translational arrest. And this IRF3 activation leads to type 1 interferon signaling. This is at least one molecular pathway that explains why proteasome mutations lead to an interferon signature and an interferonopathy in patients. And I would like to thank my collaborators. I have mentioned all of them. The group of Nantes, where we collaborate a lot, is also to be mentioned. And the others I think I have mentioned during the talk, and I also would like to thank my funding agencies. This is mainly funding by the German Research Foundation, and I thank all of you for attentions, and I'm now happy to take questions, and I'm sorry that I'm a little bit late.
Thank you very much. Thank you for your very interesting talk, and also a huge molecular patterns in this pathway. So sophisticated. Thank you very much. I'm waiting for the questions. I should thank all participants for joining us. Before asking any question, I want to ask the first one. As you know, the primary immunodeficiencies has such point mutations like Mendelian susceptibility to mycobacterium diseases. As rare diseases, we have some kind of these patients in our center as a referral center for primary deficiency disorders. We have some problems for interferon gamma or interferon gamma receptor deficiency in them. And we go through the treatments like interferon gamma or somehow stem cell transplantation for them. So I want to ask, as these two diseases has extreme patterns at the first and at the end of a spectrum, is there any similarity for treatments like stem cell transplantation for interferonopathies or maybe gene editing using CRISPR assay? I want to ask about that. Thank you.
Yeah, as far as I know, there is at the moment no trial for genetic engineering in these type of type 1 interferonopathies. The stem cell transplantation at least works in these 3 patients I have presented. And I think there are a couple of others which also where the publications came out this year or last year that I'm aware of. I know that Elke Krüger has tried to treat Aicardi-Goutier patients with inhibitors of reverse transcriptase, so antiviral therapy, if you want, with kind of positive effects, and he is trying to proceed with these trials. But this is all of these treatments are at the stage of clinical trials. As far as I'm aware of, to me, I'm not aware of any trial using a CRISPR-based method to go into the stem cell transplant. They used donors which are compatible.
So what is the routine treatment protocols for this kind of patients, and monoclonal antibodies do you use?
You mean for type 1 interferonopathies?
Yes.
The JAK inhibitors. So you have these Janus kinase inhibitors which are on the market now. There was a positive clinical trial run by Rafaela Goldbach-Mansky at the NIH in the US. The PRAAS patients are quite responsive to this treatment. And I know from colleagues in Germany that they also try to use that very successfully. I'm aware of one patient which was treated with this anifrolumab, which is this inhibitor or this monoclonal antibody against the IFN receptor. This study, yeah, I hope that we are involved in the study, and I hope that will come out soon. That this is also a kind of treatment option, but I'm not aware that these treatments are routine things. So for SLE, they are starting now routine trials.
Thank you very much. We are waiting for the questions from participants, and also if somebody want to talk, we can open his microphone. Another question, as a lab woman, I want to ask about the diagnostic protocols. If we have not microarrays or some sophisticated techniques that you said about that, can we use ELISA methods? I know that intracellular cytokine assay would be more accurate, but can we use cytokine assay for screening the patients at the first?
So I think if you have a flow cytometry system, this would be the most easiest way to screen patients for interferons using this cyclic antibody. There is this publication we were part of, but there are also a couple of others which you can read protocols about that. This is quite simple. Here you have to make sure that the blood is quite fresh.
Yeah.
And for the easiest method is the use of a qPCR, where you amplify this 6-gene panel of the ISGs, and this is based on the method of Rice et al. In 2017, as far as I remember. This is a Jakes Met paper from the Crow group.
Yeah, thank you very much. And my last question is about the COVID patient, the patient with interferon type 1 interferonopathies who are suffer from the COVID-19. How they respond to this kind of viruses, as they have lots of interferon production and can protect them from the viruses. Can they have very milder than the healthy controls?
Yeah, actually, we have here ultra rare diseases, so we don't have many of these patients, and there are no big trials to investigate this. I can say that there is at the moment no sign that vaccinations are somehow lead to overproduction or overreaction in these patients. There was a recent publication also by Rafaela Goldbach-Mansky, who stated that these autoinflammatory patients can be vaccinated regularly. And from the infection, I'm not aware of any study which was investigating this special case. I think that the hyperproduction of interferons should protect from viruses. I'm aware of clinical observations that the clinicians or the pediatricians observe that these patients do have less viral infections in their course of life. However, in some cases, these diseases also cause kind of immunodeficiency. And this is due to or dysregulation of differentiation in the hematopoietic system mainly, and they suffer from leukopenias. And here this might be tricky and maybe also worsen if you have such a patient. But I'm not aware, and I'm not a clinician to answer this question, but this is my thinking about that and what I know about it.
Thank you very much. And my last question is about the prevalence. Do you have any data about the prevalence of this kind of patients in the world or maybe in the Europe?
Not really. So I can say that we have a couple of them, and the improvement of genetics using NGS somehow makes us or allows us to identify these patients also now from databases. And for the proteasomopathies, I think we have about 100 patients across the world. I do not know numbers of Aicardi-Gautier, but this is already more. At least the patients we know are more than compared to proteasomopathy patients. But this might be due that the disease was discovered earlier. So I think the progress in understanding of these diseases will also bring us to higher identification rates.
Yeah, maybe we misdiagnose them or cannot.
Yeah, that's the case.
Yes, thank you very much. Thank you.
You're welcome.
Very interesting talk. Thank you. Dr.
Yes. Yes, thank you very much. Thank you, Dr. Krüger, for this very informative, very interesting presentation. The interferonopathies are actually very recently introduced. So much is still to be known. And you presented such a fabulous lecture from the pathophysiology to manifestations, to diagnostic methods, and to treatment. And we learned a lot. I'm very grateful for your presence here. We are honored to have you here. And is there anything else you want to add?
So I see a hint. There is obviously a question.
No, actually,
Dr. Salehzadeh, I think he wants to have it.
Yeah, of course, yeah. Please. Dr. Salehzadeh is actually a pediatric rheumatologist at Ardebili University of Medical Sciences. He is now living in Australia, and he has had marvelous contributions to pediatric rheumatology, a very fantastic register system for FMF patients, because he has covered so many FMF patients in that region. In Ardebili, we have many patients of FMF, and we're honored to have him here. Please, Dr. Salehzadeh. Can you?
You have to unmute him, I think.
Yeah, but I think he has access to the microphone.
Yeah, thank you.
Yeah, please.
Thank you. Please activate my micro. Thank you very much. Thank you, colleagues, and thank you, Elke. Very excellent presentation based on the proteasome roles in autoinflammatory disorders. Details, very delicate details, of course. Thank you very much. It was very useful for me, of course, all of my colleagues and audience. I have a question about the rheumatologic manifestation of neoplastic disorder. We know in neoplasms we have malfunction cells. They produce many malfunctions, proteins, and many products that are not normal in structures and functions. What is the role of proteasome as a dysfunction in rheumatologic manifestation of neoplastic disorder?
Yeah, this is an excellent.
Is your question? Is your answer to my question is yes? It has a role. It should be a good point that we have autoinflammatory presentation in neoplastic disorder, not autoimmune presentation. Thank you very much.
Yeah, this is an excellent question. And actually, we are also looking at neoplastic disorders at the moment, together with our hemato-oncology department here. The answer is yes. Also, tumor cells in general highly rely on a good working proteostatic system. So mutations which occur during neoplastic transformation and also during inflammation, of course, they contribute to this burden of misfolded and misfunctional proteins which have to be removed. Otherwise, cells would start programs of cell death. And during hematopoiesis, this is obviously important. We are currently trying to investigate this in more detail. We do see misregulation of T cells also in our proteasomopathy samples. But when we investigate tumor cell lines, and you know that proteasome inhibitors are known treatment options in multiple myeloma, where you have misregulation of the B cell compartment. This, when we treat myeloma cells with proteasome inhibitors, we do see interferon signatures. So this is clearly involved, and this is obviously a normal cellular response to proteasome impairment, what we observe here. And the answer is yes, the Ubiquitin-Proteasome system is clearly involved in that, and therefore treatment or inhibiting proteasomes as a treatment option in hematopoietic cancer is already applied. I hope that this kind of answered your question.
Thank you very much. That was great. Thank you.
Does Dr. Salihzadeh want to add something? Dr. Salihzadeh? I hope we have him still. Wait, I will try to maybe. Oh,
Thank you very much.
Yeah.
Okay. That was a good answer. Okay. Do you have my voice? Thank you very much. Good answer to my questions. My second question is about the animal models of proteasomal disorder. Any animal models do you know about the proteasomal disorders? Some kinds of proteasomal disorders goes to neurodevelopmental, but we don't have any neurodevelopmental structures like human in animals. Do you have any proteasomal, do you know any proteasomal disorders or neurodegenerative disorders, NDD, in animals?
So we have the problem that mouse are not human, and that's the real question here. I have to answer this in a little bit more detail. So the proteasome itself is normally an essential enzyme. If you delete proteasome subunits, mice will die. This is not true for the immunoproteasome, because this is a kind of backup system. You have this is an isoform which is expressed in the hematopoietic system. So these mice are known for years. They are available. However, these are classical knockouts, and we are currently publishing a model which is a conditional model, which can be useful to the directed knockout in different tissues and cell types. There are other models available knocking out ATPases of the proteasome, so the PSMC subunits, and these mouse models suffer from neurodegeneration and neuroinflammation. But these mice are severely sick, and this is a problem with the animal facilities to have this animal burden here, because you know, this is kind of simulating human disease in the course of the neurodevelopmental disorder. However, this does not simulate the autoinflammatory disorder very well. We are currently constructing a mouse model, and there's one available from a Japanese group published recently, where there is a point mutation which is dominant negative. So there is a point mutation in the PSMB9 subunit, and we are trying to get this mouse model, and we are constructing another mouse model with the point mutation in PSMB8 to better mimic the autoinflammatory disorders. And for neurodegeneration, of course, these mice do not get that old than we get. So we have to somehow trigger the neurodegeneration by introduction of mutated proteins into the mouse. This is done for Alzheimer's disease. This is done for Parkinson's disease. But again, all these diseases are also coupled to neuroinflammation and interferon production in the brain. This is something which is known.
Thank you very much. I see that Dr. Salazade has written here in the chat room. He has thanked you, and he has said that it was a good point in rheumatologic manifestations in neoplasm. Thank you very much.
I have a question, Dr. Hasnashari. Would you use the germ-free environment for mice studies?
No, not really. Of course, we use SPF conditions. So this is, yeah, not germ-free, but you have less bacterial or microflora in these mice. Actually, to mimic the human system, it would be nice to have a humanized mouse with a micro environment which is close to the human microflora.
Yes.
But we do not use it at the moment. We use classical SPF conditions.
Yes, thank you very much. Thank you, Dr. Krüger, for your very interesting talk. I think we don't have any more questions. I'll check it now. Thank you very much. Dr. Hasnashari.
Yes, thank you. Thank you, Dr. Krüger, for your presence today. We are honored to have had you here today, and Dr. Nurizadeh for being the great moderator of today's session and for the great discussion that you had. If there is anything else, Dr. Krüger, you would want to add?
I thank you for the opportunity to talk to you, to give this lesson here. I hope that I could give a kind of overview in the molecular details of these diseases. And it was my pleasure, and I'm honored. Thank you.
Thank you very much. We are
and of course, just one additional point. If you have patients with proteasome lesions and need to investigate this in more detail, do not hesitate to contact me. And if there are any further questions, please send me an email. So it's fine to help around that.
That's great. It's a great opportunity. It's a great opportunity. Yes, of course.
I realized that in subtitle, proteasome has been written in proteasome. Please take notice this point. In subtitle, proteasome has been written in proteasome. Please note this point. Notice this point.
Of course.
Take this point. Thank you very much.
Thank you. Thank you for your always precious notes. Thank you very much. We are really lucky to have Dr. Salazade in our team, and he's so much careful. And I think, do we have another question? No. Okay. So everything is okay. So if we're done with the questions and the discussions, I would like to thank you again, Dr. Krüger. It has been such a great opportunity to collaborate with you, and the proposal of introducing the patients to your team, that's really it's a great opportunity for us. Thank you very much. And Dr. Nurizadeh, I'm very thankful for helping us today with this great session. And thank you very much. I'm grateful for participation of all you academics in this session of How I Treat Autoinflammatory Disorders course, and I hope you have had an enjoyable time. As I mentioned before, we will share the posters later with the participants, and then we can send a certificate. And we invite you to our next session, which is going to be held next Friday, entitled as PFAFA syndrome in children. And our speaker will be Dr. Susan Shenoy from USA. You can find the details on the next session on our website. Also, we will be very grateful if you can fill in the feedback form we shared in the chat forum. And thank you very much, and have a great day. Thank you, Dr. Krüger, and thank you, Dr. Nurizadeh. We will see you soon, hopefully. Goodbye.
Goodbye.
Bye.